to presenter view here, sorry. So yeah, so Satya was going to present, but Satya is a DVM PhD student, and unfortunately he's in class today doing his veterinary training. So he asked me to present, and so I came to Michigan State about 6 years ago. Prior to that, I'd been at Stanford for 27 years, and when I came to Michigan State, I decided to start a new project that I've been thinking about for some time, and that is to engineer endosymbionts for cellular control, and ask if we could recapitulate eukaryotic evolution and create novel organelles and get them to communicate with the host cell in a way that we could direct host cells to do things that would be either beneficial for health or at least interesting from a scientific perspective. So basing this on the endosymbiont theory, we thought, well, would we be able to start with the mitochondria itself and try to engineer it in the cell and then transfer it from cell to cell, but without having autonomous replication made it quite challenging. And so we reasoned that we could do this with otherwise free-living bacteria and convert them into engineered endosymbionts, and begin to develop a symbiotic relationship. Work has been done on E. coli and yeast to get them to be form a symbiotic relationship where they would exchange nutrients, and we'll work towards that as we move forward. So the idea would be that we would take otherwise free-living bacteria and try to get them inside the cell. And we've done this in a number of different ways. We tried with cationic lipids in a process we call endofection, but we decided we would start with the easiest possible targets, so we started targeting macrophages. To do that, we would need to get the, once we got the macrophage to phagocytize the bacterium, we'd use Listeria lysino from Listeria monocytogenes to escape the endosome so that the bacteria would be in the cytoplasm. We then would engineer the bacteria to make a protein that would be then secreted from the bacteria into the cytoplasm, and with nuclear localization signals get that into the nucleus, with the idea that if we encoded eukaryotic transcription factors in this engineered endosymbiont, that we could get them to interact with pathways in the cell and have the cell change its physiology in a direction desirable for therapeutics or just for interesting directions of cell biology. Once we accomplish that, then we would work towards engineering codependence for symbiosis to try to retain the bacteria for longer periods of time inside the cell. So when we were thinking about chassis organisms to begin these studies, we wanted a well annotated sequence genome that had a well-developed genetic system and characterized transporter that would get the genes out of or the proteins out of the bacterium into the cytoplasm. We want something non-pathogenic, and we could learn a lot from bacterial therapy. For example, BCG is the standard of care for bladder cancer, so we started working on ideas where we could then use this as a cancer therapy. We started with Bacillus subtilis for the reasons that I just described, and using the TAT transport system to get folded proteins out of the bacteria into the cytoplasm of the cell. So the idea would be get a reasonable number of cells, bacterial cells inside the cytoplasm that then could communicate with the genome. So I'd like to go through that scheme step by step. The first was to demonstrate that the bacteria were internalized into these macrophage cell lines. We've done this with various cell lines, but also now with primary macrophages, and that they would escape the endosome by staining with LAMP1. We could show that the bacteria were in the cytoplasm. They would survive in the cytoplasm. We could see them divide from 2 to 3 cells to a dozen cells. And even in the case of Bacillus with a dozen bacteria per cell, that the macrophage viability was still pretty good. We call this multiplicity of infection, but it's not really an infection because these are non-pathogenic organisms, so it's really multiplicity of endofection, and ratios of 10 bacteria to 1 macrophage, or 25 to 1, or 50 to 1, gave reasonably good viability, and we could get the bacteria and the co-cultures, essentially bacteria and macrophages, to survive for 24 to 48 hours. We then needed to demonstrate that we could get a bacterial protein expressed, secreted, and then targeted to the nucleus. To do this, we used the mannose-regulated promoter to drive expression of beta-galactosidase, and we put it into these cells either with a nuclear localization signal or without. So most of this is showing the controls. But if you induce with mannose and you have a nuclear localization signal, you see the beta-galactosidase in the nucleus, and you could look at the differences here with all the different controls, showing that we could induce a protein, we could get it into the cytoplasm, and then transport it into the nucleus. So from there, we started making bacterial operons comprised of mammalian transcription factors. We started with transcription factors that would affect macrophage biology. So macrophages are typically M0, and they can be polarized to M1, or which would be pro-inflammatory, or an M2, which would be anti-inflammatory. These transcription factors are thought to play a role in that, and we would drive these with the mannose-inducible promoter. We call this our SK operon for STAT and CLIF6, and that drives this M1 phenotype to be pro-inflammatory. You can imagine that in both cancer and in chronic inflammation, the vasculature is leaky. So if any bacteria you inject IV into a mammal will accumulate in a tumor because of the leaky vasculature and then be consumed by macrophages. And if you could convert that anti-inflammatory macrophage to a pro-inflammatory macrophage, you could turn a cold tumor into a hot tumor. Similarly, in chronic inflammation, autoimmunity, arthritis, if you had this KG or the CLIF4 GATA operon, you would drive an M2, which would be anti-inflammatory. Chronic inflammation also has this leaky vasculature, so the bacteria would accumulate in sites of inflammation, be taken up by macrophages, and drive then an M2 phenotype. So these, again, with all the controls, the M1 and M2 as the standard for driving these polarization, and then the LLO bacillus with either SK plus and minus mannose or KG plus and minus mannose. If you look then at the cytokines that are secreted from these macrophages, bacillus does increase cytokine production, as you might expect. But if you look at the KG inducing the M2 transcription, or make the KG transcription factors inducing an M2 response, we see these cytokines induced over just the bacteria alone. So it looks as though our transcription factors are getting to the nucleus, and we've begun to stain these cells to demonstrate that's indeed the case. So where do we go from this? The macrophages and the macrophage polarization was really just a proof of concept to show that we could get bacteria to change the host cell phenotype. Where we'd like to go with this is towards reprogramming, where we would make a Yamanaka operon so that we could take differentiated cells and drive them towards an iPSC, and then flip another genetic switch in the endosymbiont and drive those iPSCs towards a cell type of a desired cell type. You can imagine injecting these bacteria IV, having them taken up in the Kupffer cells in the liver, flip a switch to make a Kupffer cell into an iPSC, and then another genetic switch to drive hepatocyte differentiation, to drive essentially tissue regeneration from outside the body by creating these bacteria to program cells back into hepatocytes. We reasoned that this would be a reasonable target because we've been studying stem cells for some time. In this case, we could take a single hematopoietic stem cell responsible for all the blood types in the body. We could put that into a lethally irradiated animal, watch it engraft, and then watch that single hematopoietic stem cell repopulate the entire hematopoietic system over a period of 31 days using luciferase as a reporter. What we reasoned with the engineered endosymbiont approach is that if it was very inefficient, which in macrophages it's not bad, but if it was in stem cells, it would probably be relatively inefficient, we could then drive a single cell to repopulate an entire tissue, in this case, the blood. So we could tolerate those inefficiencies of engineering by having a small number of cells being able to reproduce a large number in the end. The challenge we'll be faced with, of course, is to keep that endosymbiont in the cells as they divide. So we've been looking at mitochondrial proteins to put on the surface of the bacterium so that when the cell divides, we would pull the bacteria into the two daughter cells. So we've made a series of operons, including the Yamanaka operon for iPSC reprogramming. There's a series of transcription factors that would make those iPSCs into cardiomyocytes, hepatocytes, pancreatic beta cells, skeletal stem cells, or neurons, and begun to engineer these operons. This one we're testing now for the reprogramming. Of course, macrophages are notoriously difficult to reprogram, but we'll work with these to see if we can get those to reprogram appropriately. The challenge we have, I see that I have the WMIC World Molecular Imaging Congress logo on the bottom. I presented this last week in Prague. So one of the things we'd like to do is be able to control these from outside the body and not be limited to using sugars. We've been looking at xylose promoters and acetyling xylose so that it does get into mammalian cells. We've been looking at mannose and, of course, galactose-inducible promoters. We'd like to do this with magnetic control. So we've been looking at magnetotactic bacteria as another chassis organism. Magnetotactic bacteria make these iron oxide particles, usually decorating the inside of the cell in this linear fashion so that the cells, wild-type cells, can negotiate using Earth's magnetic fields to travel through the water column to find optimal oxygen and nutrients. We started working with a MSR1 mutant of this magnetospirillum from Dirk Schuler. We like this because the magnetic particles are all in one cluster. That makes them easier to image by magnetic particle imaging. Magnetic particle imaging is a way of looking at the magnetic properties of particles, and so you get a bright spot on a dark background. We also engineer these bugs to express the Lux A through E operon, the modified luciferase operon from Photorhabdus luminescens, so that we can look at the bugs glowing in the dark. We can look at the magnetic properties. And by alternating magnetic fields, we can warm up these magnetic particles, and we can activate thermally regulated genetic systems. So this shows that what those magnetotactic bacteria look like in animals. They glow in the dark, so after injection you see them all over the body. They slowly get cleared over time. You can image them by magnetic particle imaging, and you can superimpose those images on a CT scan, and you can see that these bacteria accumulate in the liver, which is why we think the Kupffer cells are such a good target. These images look different because this one's surface weighted, because optics, the light doesn't go through tissues. This is not weighted by any surface measure, so you can see as they accumulate in the tissue, but you can't see the weak signals from distributed bacteria throughout the body. So we can track these bugs. We can ask how long they last, how long they last in the liver, where they go. We can put fluorescent tags in them and look at them in the cells in the liver. We can also demonstrate that we could melt off temperature-sensitive repressors from operators and turn on luciferase signals using an alternating magnetic field generator that was built as a partnership with Magnetic Insight. And here's an example of that, just these magnetotactic bacteria in a 96-well plate on a thermal cycler, showing that with time and temperature, that you can turn the Lux operon on, and then we can do the same thing with alternating magnetic fields. If you warm them up with a magnetic field, you turn on the Lux operon relative to the unheated cell. So with that, I'd like to thank everybody we've worked with. The LLO is from Dan Portnoy. We've been working with Caleb Bell and Rash Kameli on the magnetotactic bacteria. Just started this partnership with John to try to see if we can do similar things with the minimal genome bug. And these are the people that have been working on this project in my group currently, and past members of the group. So with that, I'll stop and take any questions. Thanks.
Chris, so what do you think the biggest challenge to getting JCVI, to getting our minimal cell to work, given its sort of low metabolic rate relative to some of these other organisms?
Well, I actually like the low metabolic rate a lot because one of the problems we have in some macrophages is that the bacteria grow to the point where they actually lyse the macrophage because they're no longer in the endosomal compartment.
Mm-hmm.
And so I like the slow growth rate for that reason. And I'd like to match the bacterial replication to the cell division of the macrophage. Now, when you get into stem cells, they divide fairly rapidly, but generally, 8 hours. So if I can get that match, then when the cells divide, I could have the daughter cells each containing half the number of bacteria. So the biggest challenge, so getting them to replicate at the right rate. It turns out that if we take primary macrophages from the body and we do the same studies, we get the opposite effect. We get the macrophage is still able to kill the bacteria as opposed to the macrophage cell lines, which tend to let the bacteria replicate. And so we're beginning to look at ways to affect the autophagy pathways in the host cell of the primary macrophage so that the bacteria will persist. The biggest challenge is getting them to persist, and getting that symbiotic relationship to be tolerated.
Well, you saw Daniela Bittencourt's data yesterday suggesting that the macrophages may never even see these cells. Or maybe not, but
I missed it yesterday. I had to leave a little bit early, but yeah, so when you say won't even see them, I mean, it won't recognize them or acknowledge the presence. What do you mean by won't see them?
So when we have incubated JCVI Syn3B with phagocytizing cells, they are ignored completely, unlike the wild type organism.
Yeah, we've repeated some of those, and we see the exact same thing. And so we think that we may be able to facilitate that uptake, and that's why we started using some of the cationic lipids for directed uptake to put some signal on the surface of the Syn3 that would allow the macrophages to take them up. So another way we've been looking at is just to spin them down into the macrophages to try to physically drive the contact. And
you've got those cells that the macrophages see now in your hands. Yeah. I think as of maybe yesterday or it
was last week. We received it last week. So yeah, thank you for sending those. Yeah, we'll look at those as well. And Ahmed and Tony have begun growing them and begun to look at them. So yeah, hopefully we'll make some progress there.
Other questions for Chris?
I just have a comment. So here is Bogumił Karaś from Western University in Canada. We've been doing a little bit of work with different bacteria trying to make similar things like the E. coli yeast chimeras. So I just wonder if you would be at all interested. We have this Haemophilus influenza with very small genome with number of different fluorescent genes, or E. coli plasma leishmaniae, which is similar to the mycoplasma, but they are different. They're also small. We also show that they can be like really nicely engulfed by yeast cells and other things, but they don't require serum. So essentially, I wonder if you'd be at all interested in doing some quick check with either Haemophilus influenza or E. coli plasma leishmaniae, if you could provide like fluorescent strain. So that was just more of a comment then.
Yeah, that'd be fantastic. I think we're always looking for the ideal chassis, and of course, minimal genome is preferred for us because we'd like to not be creating new pathogens, and the smaller the genome, the less likely they are to have any pathogenic capability. The things we're introducing, like internalins and LLO and adhesion molecules, are all from lessons learned from pathogenic organisms. And so I'd be very interested in looking at some of your strains. I think would be great. We'd look at them in both macrophage cell lines and in primary